Alright, hi guys. We are continuing with chapter 3. We are working on section 5, nucleic acids. Nucleic acids are a polymer of nucleotides. That's the base monomer unit. There are two different varieties of nucleic acids. DNA, which is deoxyribonucleic acid, which is the genetic material that stores information for its own replication and for the sequence of amino acids in proteins. So basically they are the blueprint for all of our cells. Then our other category is RNA, or ribonucleic acid. They perform a wide range of functions within cells, which include protein synthesis, or the building of protein, and the regulation of gene expression, which we're going to talk about soon, or at some point this year. I'm not exactly sure about the pacing. We'll get there, don't worry. The structure of a nucleotide. Each nucleotide has 3 parts. The first part is a phosphate group, a pentose sugar, pentose meaning 5 carbons, and a nitrogen containing or nitrogenous base. There are 5 different kinds of nucleotides found in nucleic acids. There are 4 types found in DNA. They are adenine, guanine, cytosine, and thymine. RNA contains 3 of the same ones, adenine, guanine, cytosine, but they contain something called uracil rather than thymine. These nucleotides are joined together by a series of dehydration synthesis reactions. Remember, that's whenever we take a water molecule out and force 2 monomers together. They form a linear molecule called a strand, which is just a sequence of those nucleotides bonded together. Here is a nucleotide. We have our phosphate group. We have our pentose sugar. Remember, that means 5 carbon sugar, so 1, 2, 3, 4, 5. And our nitrogen containing base over here. So those are the 3 parts. All right, the phosphate looks like this. The sugar in deoxyribose looks like this. In RNA, it looks like this. It has 1 less oxygen. Here are the nitrogen bases that can be found. They are broken down into 2 categories, pyrimidines and purines. Purines consist of adenine and guanine. Pyrimidines consist of cytosine, thymine, and uracil. Remember, thymine is only found in DNA. Uracil is only found in RNA. So here is a diagram just for your information. The structure of DNA and RNA. The backbone of the nucleic acid strand is composed of alternating sugar-phosphate molecules. RNA is predominantly a single-stranded molecule, whereas DNA is double-stranded, which means there are 2 strands held together by hydrogen bonds. And DNA is composed of 2 strands held together by hydrogen bonds between the nitrogen-containing bases. The 2 strands twist together around each other, forming what's called a double helix. The nucleotides may be in any order within the strand, but every time adenine will bond with thymine, and cytosine will bond with guanine. So there's always a purine bonded with a pyrimidine. The bonding between the nitrogen-containing bases in DNA is referred to as complementary base pairing. It's also called Chargaff's rule. And the number of adenine and guanine purines will always equal the number of thymine, cytosine, pyrimidines. Here is another handy-dandy chart. We have DNA and RNA listed. The sugar for DNA differs a little bit from RNA. DNA contains deoxyribose, whereas ribose is in RNA. The bases are a little bit different. DNA has thymine, RNA has uracil. The structure is a little bit different. DNA is double-stranded, whereas RNA is single-stranded, and DNA forms a helix or like a twisted ladder shape, whereas RNA does not. Here is a picture of RNA. You'll notice that it's a straight line. We have a phosphate bonded to a sugar, bonded to a phosphate, bonded to a sugar, bonded to a phosphate, bonded to a sugar. That is that repeating sugar-phosphate backbone. Off to the side, here we have our nitrogen-containing bases. We have cytosine, adenine, uracil, and guanine. Here we have a diagram of DNA. You'll notice that it is a twisted helix. Okay, if you look really, really closely, you can see the sugar and the phosphate bonded in that alternating sugar-phosphate backbone, and the hydrogen bonding between the nitrogen bases. Here we have cytosine and guanine. They have 3 hydrogen bonds holding them together. Right below it we have an adenine and a thymine, and they only have 2 hydrogen bonds holding them together. ATP, or adenosine triphosphate, is a nucleotide composed of adenine and ribose, which is adenosine, and 3 phosphates. That's where the triphosphate comes from. ATP is a high-energy molecule due to the presence of the last 2 unstable phosphate bonds, which are easily broken. Hydrolysis of the terminal phosphate, or the last phosphate on that molecule, yields an ADP, or adenosine diphosphate, and an inorganic phosphate. Okay, so breaking that phosphate off of the ATP and making it into ADP and a P releases energy. And that is energy to do cellular work. The hydrolysis of the ATP molecule can be coupled with chemically unfavorable reactions in the cell to allow the reactions to proceed. Example, key steps in the synthesis of carbohydrates and proteins, and muscle contraction and nerve impulse conduction. So breaking that phosphate off allows these things to happen. ATP is oftentimes referred to as the energy currency of the cell, because anytime the cell needs energy, it uses this ATP molecule. And this is what it looks like. Here is the adenosine, and here are the triphosphates. We have 1 phosphate, 2 phosphate, and 3 phosphate. This 3rd phosphate is not happy being there. It really, really wants to break away. So when it does, it takes some energy, or it releases some energy with it. And that's what I got for you guys. If you have any questions, feel free to rewind the video, shoot me an email, or ask me in class tomorrow.